Hello, my name is Alexander Nesser, and I am a third-year student at the Rochester Institute of Technology studying biomedical sciences. This summer, I participated in a research project slash co-op within Dr. Thomas's giant phage lab, which dealt with the bacteriophage SPN3US, a photo of which is displayed on screen, and the bacterium Erwinia amylovora. Erwinia amylovora is a gram-negative rod-shaped bacterium. For those who don't know, gram-negative bacteria have two membranes, an outer and an inner one, while gram-positive bacteria only have one outer membrane. It was the first bacterium shown to infect plants, causing the devastating disease Fireblight, named after the charred color it causes infected plants to turn. An example of an infected plant is shown on screen. Fireblight affects plants in the Rosaceae family, which includes apple and pear trees. This is of particular concern for farmers in states like New York, which is the second largest apple producer in the country. Due to the potential to wipe out whole orchards, the journal Molecular Plant Pathology has placed it into one of the top 10 pathogenic bacteria. Presently, E. amylovora can be found in all territories of Canada, many states in the US, and most recently Europe. The spread of this bacterium to other continents is raising alarms amongst farmers across the world, as fireblight threatens to severely reduce the global production of apples and pears. E. amylovora has typically been treated by pruning off infected limbs or with antibiotics such as streptomycin or oxytetracycline. While presently this strategy may work, the rise of antibiotic-resistant E. amylovora strains is beginning to exacerbate this problem. The continued use of antibiotics kills off less resistant bacteria, leaving those with mutations that confer a higher resistance alive and able to reproduce. This cycle is worsened with the overuse of antibiotics, as it leads to more and more resistant strains, making E. amylovora harder to combat. For this reason, we are beginning to look to alternative ways of controlling this bacterium. In the last decade, it has been discovered that some prokaryotic organisms have an adaptive immune system called the CRISPR system. CRISPR stands for the clustered, regularly interspaced, short palindromic repeats within the genome of an organism with a system. A vital part of this system is the CRISPR-associated protein, abbreviated Cas. There are a few different types of this protein, for example Cas3 and Cas9, each with its own function. Current research with the CRISPR system has revealed its potential to modify genomes. The biotechnology CRISPR-Cas9 has the ability to precisely insert, delete, or replace sections of DNA, giving genetic engineers greater control over the expression of target genes. Current studies are exploring the viability of the CRISPR-Cas9 system in the treatment of genetic diseases. Because of its ability to precisely target DNA, it may be possible to correct disease-causing mutations without side effects. However, further research with CRISPR-Cas9 in humans is delayed until its safety can be proven. The CRISPR system in bacteria is similar to our antibody-mediated immune response. However, while ours codes potential threats in antibodies, signaling for white blood cells to come and eliminate the threat, the CRISPR system detects harmful DNA sequences like those ejected by bacteriophages and eliminates them before they can do any further damage. These specific sequences are stored in their genome, and the CRISPR system knows which sequences to quote-unquote look out for due to previous exposure to them. This whole process is summarized by the graphic on screen. While some bacteria contain the necessary genes to have a CRISPR system, it may not be active. It's currently not yet known why this is the case. For Erwinia amylovora, the consensus is that its CRISPR system is active, however, it has not been conclusively proven. Salmonella phage SPN3US is a bacteriophage. A bacteriophage is a type of virus that infects bacteria, using its host replicative machinery to reproduce. SPN3US is of particular interest to us because it is a certain type of bacteriophage. It is a giant phage. The reason it is labeled a giant phage is that its genome is particularly long. It's about 240,000 base pairs long. This is drastically different from the average bacteriophage genome lengths, as an overwhelming majority fall below 80,000 base pairs. As you can see by this graph, the average genome length of a bacteriophage is well below 80,000 base pairs. SPN3US's genome is over 3 times this amount, which is considerable. While SPN3US infects Salmonella enterica, our research has shown that it is also able to infect Erwinia amylovora. This is interesting to us because it is not common for phages to jump between genera like this, especially when one of them, Salmonella, infects humans while the other, Erwinia, infects plants. SPN3US prefers motile hosts with a flagellum. Both Salmonella enterica and E. amylovora have a flagella, which can explain why it's able to infect Erwinia. Not much is currently known about the reproductive cycle of giant phages. It is currently unknown whether giant phages such as SPN3US rely on host bacteria's replicative machinery or use some of their own to reproduce. The main goals of this project are 1. To test the hypothesis that Erwinia amylovora has an active CRISPR system and that it can acquire resistance to SPN3US via this adaptive immune system, and 2. Determine which Erwinia amylovora genes are necessary for SPN3US infection. This will be done through the isolation of bacteriophage insensitive mutants, abbreviated BIMs. After streaking each BIM to clonal colonies, the efficiency of each one to resist SPN3US will be measured via an efficiency of plating test, abbreviated EOP. Once the phenotype of each BIM has been determined, they will be genetically analyzed using PCR and next-gen sequencing to determine the source of phage resistance. The E. amylovora BIMs were isolated by students in microbial viral genetics utilizing UV-treated SPN3US. While the UV-treated phage was still infectious, the treatment weakened it, allowing the bacteria to more easily become resistant to the phage. A mixture of UV-treated phage and wild-type phage was used for infection, which introduced selective pressure for the UV-treated phage. The addition of wild-type phage also eliminated any bacteria that were not resistant, making it easier to identify BIMs. This mixture was then plated on an overlay with wild-type E. amylovora. After incubation, the surviving colonies were marked and transferred to their own streak plate. In order to make it so the colonies were clonal and that no phage particles were still present, which would interfere with DNA sequencing, they were streaked out into 5 successive generations. Examples of such streak plates are shown here. Ideally, you want to streak out to single colonies. It is generally assumed that each colony started from a single bacterium, which is important as you do not want to be plating and later sequencing DNA from 2 or more different bacteria. This summer, I picked up where this project had left off. First off, new EOPs needed to be done. EOP tests are used to compare BIMs to their wild-type counterparts. An example of such comparison is shown on screen. The photo on the left is of wild-type E. amylovora, while the photo on the right is an E. amylovora BIM. The numbers represent the dilution of phage stock plated in that area. More on that later. EOPs not only verify that the BIMs are resistant to SPN3US, but also show how resistant each BIM is. BIMs are not always 100% resistant, which is why it can be crucial to conduct an EOP test, as they can be, for example, only 25% more resistant than the wild type. This test is also important because it helps us to identify BIMs that are nearly or fully resistant to the phage, meaning that they are of particular interest to us. Genetic analysis on such BIMs can give us a better understanding of which genes SPN3US needs for successful infection. Previous EOP results from 2019 were not tidy. This was because of E. amylovora, its tendency to grow quickly, making any plaques seen on EOP plates hard to identify. Furthermore, EOPs between BIMs were not consistent or comparable to each other. I needed to create a method that generated consistent EOPs. I would need to play with the plating media, the liquid culture growth conditions, and other factors to try and get clear and consistent plaquing. A specific type of SPN3US was used, which our lab calls SER. SER is SPN3US that has been selected for because of its ability to infect E. amylovora better than wild-type SPN3US. In addition to using SER, plaquing can be improved by using plates with ampicillin, an antibiotic. Ampicillin is able to weaken the bacteria, making them more susceptible to infection by our phage. To begin with EOPs, a serial dilution of an SER stock were prepared from 10 to the negative 1 to 10 to the negative 8. Pictured here is an example of how a serial dilution is made / what it is. For EOPs, 100 microliters of a one-time stock was diluted in 900 microliters of SM buffer. After vortexing, 100 microliters of this solution was then transferred to a new tube with 100 microliters of SM. This was repeated to create dilutions ranging from 10 to the negative 1 to 10 to the negative 9. An important part of our original phage stock is its titer, which is essentially its concentration. The unit of measurement for our titers is plaque-forming units, abbreviated PFU, per milliliter. It's important that the titer not be too low, as plaques will not develop well or at all. To calculate the titer, the formula on the screen is used. For example, let's say you plate 5 microliters of a 10 to the negative 8 dilution. You observe 5 plaques. Plugging these numbers in, you get 100 billion PFU per milliliter, which we consider a high titer, making it good for EOPs. For EOPs, liquid cultures of each BIM and a wild-type control were prepared. Each liquid culture was added into an overlay and then poured onto a fresh agar plate. Spots of each dilution were then added onto the overlay once it had solidified, and then the plate was incubated overnight. After incubation, resistance to the phage was verified, and the specific resistance of each phage could be visualized. Displayed here are 4 EOP tests. The numbers on each plate represent the dilution series plated in that area. For example, -7 means that 10 to the -7 dilution was plated there. Comparing the wild type Erwinia, labeled EA273, to the BIMs CJB5-7, CJB10-7, and YR4.5, you can see how the wild type plaques from -1 to -6, while the BIMs do not plaque at all. As you can see, it is very easy to visualize the resistance of each BIM. Recently, I have ironed out a way to get E. amylovora to give good results for EOPs. I tried growing E. amylovora at room temperature versus at 28 degrees Celsius overnight, plating immediately after incubation versus after being stored in the refrigerator overnight, and overnight liquid culture versus a couple-hour liquid culture. What I found was E. amylovora grown in 2 milliliters of liquid culture for a few hours, plus incubating the EOP plates at 28 degrees Celsius, was consistently better than all other conditions I tested. Research isn't always perfect, as you can see. It involves a fair bit of troubleshooting and trial and error. Now that I know how to get consistent and comparable results between EOPs, my next steps are to start conducting all new EOPs for every BIM. After this, I will analyze the genetic basis for SPN3US resistance in each BIM. DNA assembly of each BIM will need to be done de novo, as previous DNA assembly was reference-based and may have missed new CRISPR acquisitions. If you have any questions or would like to learn more about our research, feel free to email me at ajn9814@rit.edu or leave a comment on this video. I'll try to get back to any inquiries as soon as possible. Lastly, I'd like to thank Dr. Thomas, my lab partners, and the RIT faculty members that helped me throughout this project. I would not have been able to do any of this if it wasn't for their help.